Ladies and gentlemen, those who are attending double educations, this double education is providing different topics on general biology for freshmen and also in inclusive education, grade 9 up to 12 biologies. So today I have grade general biology for freshmen unit 4. So if you are new for our channels, please subscribe double educations. The presenter of this channel is Mr. Mahfouz Muhammad, the instructor of Jijiga University, Department of Biology. So please like, subscribe our channels, and also if you have any comments, suggestions, any topics, put on under the comments, and I will prepare and also support you. Please support your friends, brothers, and neighbors by sharing in your Telegram groups in different class, and also share for your friends, because if many student is served from my channels, I'm become very happy. Our goal is, our vision is long like double. Our vision is very long. We have a good visions. This channel is prepared channel for educational channel and motivational speech in different tips, studies tips, how we will become success in our futures. So I will prepare different topics. If you have any topics that I will prepare for you, put under my comments. Like my video, support me. Let me start general biology unit 4, enzymes, outlines. Under the outlines, we have study the major topics, enzymes, generally about enzymes. I hope you know what is enzymes. You study in grade 11s about enzymes, the characters of enzymes, structure of enzymes, function of enzymes, a lot of things. And also you study in grade 11s unit 5s bioenergetics and biosynthesis about respirations and photosynthesis. Bioenergetics means respirations, biosynthesis means photosynthesis. Under this we study about cellular respiration and photosynthesis. Lastly, in these units, we talk about metabolic disorders, about their diagnosis and the treatments, how we are treating these metabolic disorders. And let me start outlines or introduction by metabolisms. So, as we know, metabolism or any living thing is carried out metabolisms, because this metabolic process is the process of building up of new tissue or new molecules, and also breakdown of large molecules. So living cell is constantly active in a tremendous biochemical activity vital for life of living organism take place in a cells. Example of this cellular activities: building up of new tissue, the tissue that is making new tissue, replacement of old tissue, the dead tissue, conversion of food to energy, or converting different macronutrients or metabolites like carbohydrates, lipids, and proteins into energy. Disposal of waste materials, the waste materials that are released from our bodies, like urines, excretions, and also for reproduction or genetic process. So this substance is transport across cell membranes. It gives genetic instruction for our or transmit for our generations. So metabolisms have different importance. So generally, what is metabolisms? You know, I hope you know, or you have hints about metabolisms. So metabolism is the sum of chemical reactions, the total of chemical reactions, the sum or total of chemical reactions that take place within each cell of living organisms. The total chemical reaction or the sum of chemical reactions that found in or carried out in living organisms is called metabolisms. The reactants and the product of this chemical reaction is called metabolites. So, as we said earlier, metabolism means the total chemical reaction carried out in living cells, but the reactant that is a product of this chemical reaction is called metabolites. The reaction of this, the reactant and the product of this metabolism is called metabolites, or what we call micronutrients or macronutrients. So the major class of metabolites the major class of metabolites we have is proteins, carbohydrates, nucleotides, lipids, coenzymes, and different cofactors called metabolites, the major class of metabolites. In metabolisms, a series of chemical reactions in which the product of one reaction is in metabolisms, we have a series of chemical reactions in which product of one reaction is substrate for the next reactions. So in metabolisms, we have a series of reactions, a reaction which is continuity. So the product of one reaction is substrate for the second reaction is called metabolic pathways. We say it for that series of reactions called metabolic pathways. So these metabolisms or metabolic pathways broadly divided into two. Metabolic pathway, what we said previous or earlier, a series of chemical reactions in which product of one reaction is substrate for the next reactions. I hope you know the difference between substrate and products. So the metabolisms or metabolic pathways divided into two. The first one is catabolisms, and also the second one is anabolisms. Let me see one by one. Catabolisms or catabolic reactions is breakdown of larger molecules into smaller molecules. Breakdowning larger molecules into smaller molecules. It is convert nutrient to energy or release energy. During catabolisms, we have breakdowning large molecules and also we have releasing energies. For examples, we have breakdowning carbohydrates into glucose. We have breakdown proteins into amino acids. We have breakdowns lipids into fatty acid and glycerols. During this process, we are preparing energy by the process of respirations. The second one is anabolisms. It's called anabolic reactions or anabolic pathways. This leads to synthesis of large biomolecules from breakdown or smaller molecules. It uses energy released by catabolisms or catabolic pathway to build large molecules from smaller molecules. For example, during in anabolisms, synthesis of large biochemicals is synthesize carbohydrates from glucose, synthesize proteins from amino acids, synthesize lipids from glycerols and fatty acids. This is opposite of catabolism. Kata means breakdowning of large molecules into smaller ones. Ana means pulling up of large molecules from smaller molecules. Generally, this two is called metabolisms. It is ensuring continual flow of energy within organisms. If these two metabolism part is present, a life is continued. Life has its own continuity. Now, when we see nature of metabolites, as you know, metabolites like carbohydrates, proteins, lipids, nucleotides, metabolites. Let me see the nature of metabolites. Metabolites have two natures. The first one is hydrophilic, which means water-loving or polar in or dissolved in waters. These are soluble in aqueous solutions. Aqueous means waters, including sugars, purine in the pyrimidines, that is the nitrogen base of DNA, nucleotides, and the nucleosides, organic acids. This part is generally dissolved in waters. The second one is non-polar or hydrophobic, water-hating one. It's not dissolved in water. It is dissolved in what? Lipids. This metabolite is functioning energy storage, like starch, glycogens. It is used for what? Energy storage. It is used for energy storage. It's used for energy storage. It's used for membrane structures. It's used for signal transaction during electron transport chain or during nervous system, and so on. At the cellular level of organizations or organisms, The main chemical process of all living matters are similar but not identical, if not identical. So all living things generally have similar chemical process. Chemical reactions that take place in living cell are also similar. A chemical reaction that take place in all living systems or living organisms or all living cells similar. Well, so we have get energy from organisms that's called autotrophs that produce their own energy by the process of photosynthesis. We have also heterotrophs consuming photosynthetic plants. So green plants use sunlight to convert water and carbon dioxide to glucose and oxygen called photosynthesis. The process in our bodies or in plants that use water molecules and carbon dioxide that prepare glucose and oxygen is called photosynthesis, or it's produced by autotrophic organisms. What is the importance of sunlight as you study in grade 11s, if you remember? Well, sunlight used to have a lot of importance. Sunlight energy in photosynthesis has a major importance. It is split water molecules into hydrogen and oxygen. So the major role of sunlight is splitting or breakdowning water molecules into hydrogens, split water molecules into half of oxygens and also into 2 hydrogen molecules. It's breakdowning into 2 hydrogen molecules and into oxygen molecules, atoms. Which means dissociation of water molecules into hydrogen and hydrogen ion and 1 electron ions. Living organisms that requires energy, like human beings, reverse the process of photosynthesis called respiration. The opposite of photosynthesis in our living cell is called respirations, because we are using the products of photosynthesis like oxygens, glucose. They consume carbohydrates and other organic materials using oxygens synthesized by plants to form water molecules, carbon dioxide, and energies. Plants use hydrogen and water molecules and carbon dioxide, but animals produce water and carbon dioxide in our cells. It's opposite reactions. So if we comparing, when we study about photosynthesis and respiration, we are comparing these two reactions. When we study about photosynthesis, it uses water molecules, carbon dioxide, and sunlight energy. Sunlight energy is used to split these water molecules into hydrogen and oxygen. But when we come to respirations, respiration is used oxygen, carbohydrate, the product of photosynthesis, and also lastly produce carbon dioxide, water, and energies. Enough for about this photosynthesis and the respirations. When we come to the process that removes hydrogen atoms or containing electron from the carbohydrate and passes them to series, energy linked series of reactions. In plant, all but 2 of steps is process that convert carbon dioxide into carbohydrates. During that process or during photosynthesis, plant is convert this carbon dioxide into carbohydrates, which means carbon dioxide is add with hydrogens. From simpler starting material in animals, fungi, or bacteria. Similarly, a series of reactions take given starting material and synthesize certain molecule will be used in other synthetic pathways. But it is similar or identical among all setups. Preparation of carbohydrates, proteins, glucose in our So if the reactions have enzymes, the chemical reaction is very fast. You see the difference between the reaction with enzyme and without enzymes. The reaction without enzymes is very too slow, but the reaction with enzyme is very fast. So when we conclude from this, the role of or the function of enzyme is speed up of chemical reactions or catalyze reactions. Almost all metabolic processes in a cell need enzymes to speed up reactions, to finish the reaction at fast, to get or sustain lives. So generally, enzymes have a great role in speed up of a chemical reaction or catalyze reactions. But enzyme is proteins. It's made from proteins. Catalyze speed chemical reaction by facilitating molecular rearrangement that support cell fractionations. So, in another definition of enzyme is first, enzyme is proteins. Second, enzyme is biological catalysts. Enzymes have active sites on their bodies. This active site is a restriction region of enzyme which substrate can bind to form an enzyme substrate complex. Active site means a place where enzymes bind with substrates to form enzyme substrate complex. Enzyme substrate complex stage means intermediate stage for the reactions. Enzymes have active site on their body that speed up or bind with substrates. Enzymes are catalysts and increase speed up of chemical reaction. Generally, enzyme is a catalyst and increase speed up of chemical reaction without themselves undergoing what? Permanent chemical changes. But at the end of the reactions, enzyme is not changed by the process or by itself. The basis is the basic enzymes reaction that can be represented as follows. We have substrates that enzymes act on. We have enzymes. When enzyme and substrates combine, they form enzyme substrate complex, that is the intermediate stages. Then they produce product and enzymes. At the end of the reaction, enzyme is not altered by the reaction or changed by the reactions. Where this is represents enzymes catalyze and also else is represent substrates. The substance being changed. Substrate means changed substance, but enzyme is not changed. P is product of the reaction, which is get from or produced from substrates. Enzyme also speed up of chemical reaction by what? Lowering activation energies. This is another importance of chemical reaction. How enzymes catalyze a reaction by lowering activation energies. When everybody asking you how enzymes catalyze a reactions, yeah, we say enzymes proteins, enzymes catalysts, enzymes lowering activation energy. How it is reducing or speed up of chemical reaction? Your answer is by reducing or lowering activation energies. Maybe somebody is asking you also, what's activation energy? And also you say activation energy is a minimum amount of energy which is required to start a chemical reactions. You see the activation energy. Activation energy means the minimum amount of energy which is required to start a chemical reactions. Let me see these graphs. This energy, this is progress of the reactions. Reaction without enzymes have higher activation energy. The reaction with enzymes have lower activation energies. You see the difference between enzyme reaction with enzyme and without enzyme. If reaction is have enzyme, it's lower activation energy, the minimum amount of energy which is required to start a chemical reaction. Well, this is a graph also shows about progress of the reaction and energy, activation energy. These activation energies, the energy which starts these reactions. High energy intermediate. But if this reaction have enzymes reduce this activation energy into this. Enzyme reduce height of the energy bearers or activation energies. And then we get a product simply. This is how enzymes work by reducing or lowering activation energy. Enzymes exhibit 4 defining characters shared by all. Enzymes have 4 characters. Let me see the character of enzymes one by one. The first character of enzyme is enzyme modifies the rate of chemical reactions or speed up of rate of chemical reactions. One, as we know, enzyme is speed up of a chemical reaction, which means modify rate of chemical reactions. Second, enzymes are not consumed in the process. In the process, enzyme is not changed by itself or not consumed by the reactions or by the process. Third, enzymes are effective in extremely small amounts. Enzymes are at the end of the reactions, or the reactions, it's not changed by itself. It's not consumed by the process. The third character of enzyme is enzymes are effective in extremely small amounts. The small amount of enzyme is catalyze a lot of substrates. This is another example of enzymes. And also, enzymes do not alter equilibrium of the reaction or do not alter the speed up of or direction of the reactions. So, an important principle is that since enzymes only reduce barriers or activation energy between product and the reactants, it reduces what? Only reduce energy or barriers between products and the reactants. So enzymes always catalyze the reactions. The importance of the reaction is enzyme is what? Catalyze the reaction in both directions. It cannot drive the reaction forward or affect the equilibrium of the direction. Maybe if the reaction is forward or backward, it's not affected by the enzymes. When we see the chemical natures and the classification of enzymes, what is the chemical nature or content of enzymes look like? How we are classifying enzymes. In these topics, we see detail of chemical nature and the classification of enzymes. But please attend me, give me attention for me, and also if new for my channels, like, subscribe, and share for your friends. If you like my video, like. Okay. All known enzymes are protein with exception of RNA enzymes. All enzymes is proteins except RNA enzymes. All enzymes protein except RNA enzymes. Some enzyme may additionally contain non-protein group before their catalytic activities. Some enzymes have non-protein groups. Enzymes have protein parts and non-protein parts. Those non-protein parts is called cofactors, or a non-protein chemical compound that is bound or tightly or loosely to an enzyme. This is required for what? Catalysis of the reactions. If this non-cofactor is present in some enzymes, that enzyme is not have, we don't have catalytic properties. If that enzymes have no cofactors, it have no catalytic properties. It gives catalysis or catalytic property for that enzymes. Apoenzyme means the enzyme's protein parts or protein portion. Without it, non-protein part is inactive. They come together and they become active. This apoenzyme plus cofactors, it make hollow enzymes or full enzymes or conjugated enzymes. This hollow enzyme is called an active enzyme with its non-protein components. You see, if cofactor is combined with apoenzymes, it forms hollow enzymes or conjugated enzyme or active enzymes which have or which speed up of the chemical reactions. Let me see types of cofactors. These cofactors, as we see previous, cofactors are non-protein parts of enzymes which tightly or loosely bind with protein parts of enzymes called apoenzyme and forms hollow enzymes. The first one is coenzymes, non-protein part compounds which is bind with the protein parts loosely or by weak bonds to apoenzymes by non-covalent bonds or by weak bonds. Examples: vitamins or compound derived from vitamins called coenzymes, or generally we call organic materials, organic materials, we call organics. The second one is prosthetic groups. Prosthetic group means a non-protein compounds which tightly bind to apoenzyme by covalent bonds. It is tightly bound with apoenzymes by covalent bonds or strong bonds. Examples: minerals. The second one is metal ion activators, like potassium iron is the common example of cofactors. And also another topic of in today's enzyme can be classified based on different criteria, or we are classifying enzyme based on different criteria. We are also dividing in difference in chemical natures. The enzyme may be described as follow by the difference in chemical natures. The first one is simple enzymes. The enzyme which is made only from protein parts or polypeptides. These contain no chemical group other than amino acid residue, or protein parts. This enzyme is especially those enzymes found in digestive tracts or digestive enzymes like pepsins, trypsin are a common example of simple enzymes, common examples of simple enzymes. The second one is conjugated enzymes. Conjugate enzyme means it is an enzyme which formed from two parts. It's made from two parts, protein part called apoenzymes, flavor protein, and the non-protein parts called cofactors. Generally, this enzyme is called conjugated enzymes or hollow enzymes. It can be enzymatic activity. It have active catalytic properties only when both present together. When two these protein and non-protein parties present together, enzyme have enzymatic activity, have catalytic properties. If the cofactor is firmly bound to apoenzymes, it is called firmly. If this cofactor is firmly bound, which means bound by strong bond, is called prosthetic groups. If instead of being more or less permanently bound to apoenzymes, that cofactor is called coenzymes. If it is bound firmly, prosthetic groups, it is bound on by the permanent or temporary or loosely, it's called coenzymes. The third one is metalloenzymes. This metalloenzymes means the enzymes or the metal cofactors involved in enzymatic reactions are maybe monovalent, have one ions or one valence, divalent or dications have two ions. Divalent, one ion is called di means two, mono means one. So the enzymes have metal cofactors in the enzyme part is called metalloenzymes. The last one is isoenzymes. A substrate may be acted upon by a number of variants of an enzymes producing the same product. Different enzyme, if different enzyme is working on the same substrate, it's called isoenzymes. The enzymes working in different substrates to produce the same products. It's called isoenzymes. You see, substrate may be acted upon by a number of variants of an enzyme producing some products. One enzyme working on different substrates called it produces some product is called isozymes. If we see the chemical nature of these enzymes, we have these enzymes simple proteins, simple only protein parts, conjugated enzymes, or complex or holoenzymes, which means protein part and non-protein part. Non-protein part means cofactors, protein part means haploenzymes. This is simple enzymes or have only protein parts. We classify these complex enzymes into two, haploenzymes and also cofactors. This haploenzyme means protein parts only, cofactor means prosthetic groups and coenzymes. These prosthetic groups have usually inorganic molecules or atoms. It usually tightly bound to haploenzymes, which means tightly attached with a strong bonds. When we come to coenzymes, this coenzyme is large organic molecules which is loosely bound to haploenzymes, which means bind with enzymes by weak bonds. Loosely bound to haploenzymes, it means bind with enzymes by weak bonds. On the basis of the substrate they act upon enzyme can be classified as follows. In previous topic, we classified enzyme based on the chemical nature of the enzymes. Now we are classifying enzyme based on substrates they act upon, the substrates they catalyze. We classify into 6 groups. The first one is oxidoreductase class of enzymes, which catalyze oxidation and reduction reactions. We classify this enzyme based on substrates they act on or catalyze. The first one is oxidoreductase, which catalyze oxidation and reduction, which means catalyze of transfer of electrons. The second one is transferases, which is transfer a group of functional groups like amine groups, phosphate group, different groups, which transfer the functional groups. The second one is hydrolysis, which means catalyze hydrolysis reactions, a cleavage of bond by the addition of waters, cleavage of bonds by the addition of waters. So they break down bonds by hydrolysis, which means break down by waters. The fourth one is lysis, which means non-hydrolytic cleavage or breakdown by without waters, cleaving or splitting a molecule by non-hydrolytic process or by the absence of waters. Lysis, breakdown molecules. But when we come to isomerization, isomerase is the fifth example of class of these enzymes, isomerases, which is isomerizing or rearrangement of molecules or atoms to form isomers. It's the arrangement of atoms to form isomers. Example is isomerase enzymes. The last one is ligase enzymes, which means joining two molecules. Ligase using energy from ATP and also formation of carbon and other bonds with energy from ATP. It form or produce what? New bonds, other bonds. It is called generally ligase. If somebody asks a question or if you are asked in the exam from the class of enzyme, which one is joining carbon molecules and form new bonds between molecules? A. Ligase, B. Lysis, C. Isomerase, and D. All. If you say the answer is ligase enzymes, you get the answers. If you are asked as from the following class of enzymes, which one is break down a bonds by addition of waters, your answer is must be hydrolysis. From the following class of enzymes, which one is cleavage or breakdown of bonds in the absence or without water molecules? A. transferases, B. oxidoreductase, C. liases, D. ligase. If your answer is C. liases, you got the answer. So please understand like this. So another major topic of today is also mechanism of enzyme actions. Enzymes have different mechanisms. Enzymes have different systematic way of working on reactions as substrates. So mechanism of enzyme catalysts vary, but all enzymes speed up of a chemical reaction by lowering energy bearers or activation energies. Energy bearers, what is lowering? By lowering activation energy bearers, separating reactant from the product. You see, separate reactant from the product, or separate product from the reactants. Lowering or reduction of activation energy may increase fraction of reactant molecules that can overcome energy barriers and also form products. So enzymes reduce activation energy to fraction of reactant molecules that can overcome energy barriers and forms a good product. When enzymes catalyze a reactions or speed up of a chemical reactions, enzymes attract substrate to its active site. So when the enzymes catalyze a chemical reactions, the active site of enzyme is attracted by substrate and they bind together. Catalyze a chemical reaction by which product is formed. Enzyme allow product to dissociate. Then lastly, enzyme is dissociate with products, and at the end of the product is enzyme is not changed by the process. The combination formed by enzyme and its substrate is called enzyme substrate complex. Firstly, when enzyme and the substrates bind together, it forms enzyme substrate complex. Enzyme substrate what? Complex, one of the intermediate reactions. Then it forms product and the enzyme is dissociated from the products, which means at the end of the product, enzyme is not changed by itself, and we are using again and again and again. It's not changed by itself. Two models have been described on mechanism of enzyme actions. We have two models which is describe about mechanism of enzyme actions, or how enzyme is worked. How enzyme is worked. The first one is look at the key models. The second one is induced fit models. Let me see one by one. Okay, these two models talk and describe about mechanism of enzyme actions. How enzyme is bind with its substrate and they also catalyze reactions. And also they talk about how enzyme is hindered or inhibited by inhibitors. The first model is look at the key model of enzyme mechanisms. This model is postulated in 1894 by Emil Fischer's German biologists. So as this Emil Fischer's enzymes have a rigid three-dimensional body contents of active sites. The three dimension of this enzyme is called active sites. As the description of this, look in the key models. Enzyme have what? 3 dimensions, 3D structures. In this analogy or similar with that, the look is enzymes. The look we have using for our house is enzyme, and the key is a substrate that we have opened for our houses. Only the correct size key, substrate into the key hole is active site of the look enzymes. So substrate is key and enzyme is log. Substrate fit into a hole of active site of enzymes, all called log. So generally they fit like log and the key what we are using in for closing our house and opening for our house. When I wanted to open the other people's house, my key is not fit to that locks. Due to that reason, it's not complementary. Only the corrected shaped key opens a particular lock. Corrected shaped keys open particular locks. So only enzyme and the substrate is complementary or feed each others or bind with each others. Substrate is binding to the enzymes is facilitated by amino acid functional group in the active sites. Substrate is binding to the enzyme is facilitated by amino acid functional group in the active site. Substrate is binding with enzyme active sites. This structure shows this. This is substrates and this is active site of the enzymes. They fit like look in the key. Enzyme is lock and the substrate is key. So when we're summarizing this look in the key models, enzyme and the active site is complementary to each others. It feeds like us look in the key models as you see in these pictures. Substrates bind with enzymes like look and the substrates like K, they form enzyme substrate complex or intermediate reactions. You see, look in the key pictures. This is a look, this is key. Look is substrate, key is enzymes, functional shapes. It is complementary with the opening site of the substrates. Opening site of substrates. It is complementary with each others. This is talk about how looking at the key model is define enzyme mechanisms, how enzymes catalyze substrates, how enzymes bind with the substrates. I hope it is clear for you. When we pass to the second models, induced feed models. Induced, which means it make change, flexible, flex the active site of enzymes. In these models, the enzyme structure is not rigid, it is flexible. But according to look in the key model, enzyme structure is three-dimensional, rigid three-dimensional structure. But according to induced feed model, enzyme structure is not rigid, but it is flexible. It changes its shapes. The enzyme and its active sites adjust shape. Even if the shape is not complementary, it is adjust a shapes, enzymes, in order to bind to substrates. If one enzyme is going to bind to substrates, if the shape is not complementary, it adjusts itself to bind with the substrate specificity. Therefore, the transition state or configuration shape will be formed. This improves catalytic reaction during the reactions. It makes enzyme active sites under tensions. This term is induced feed meaning that the precise orientation of enzymes required for catalytic activity can be induced by binding of substrates. It must be rearrange its shapes and also bind with substrate, and the reaction is speed up of the reaction. Certain substrate can bind to enzyme at site other than active site, which means inhibitors. A substance or certain substrate that bind with enzymes in order to inhibit the enzyme active or enzyme catalytic properties. In these defeat models, you see this structure is not similar with this. Then, if the substrate is close to the enzyme is change its shape. So due to these reasons, the enzyme is flexible. It's change, not rigid. It's become form conformational changes, make changes, conformational changes. Induced feed substrates change enzymes forms or shapes once in active sites. They change the enzyme active sites. So another topic is factors affecting enzyme activities. So which factor is affecting enzyme activity or the rate of the reaction of the enzymes? The activity of enzyme is affected by its environmental condition. Different conditions in the environment, such as temperatures and the pHs. Enzyme activity have different environmental factors like temperature, pH, enzyme concentration, substrate concentration, and also inhibitors. Changing this alter rate of reaction by caused by enzymes. It decrease or alter the rate of the reaction. What is the rate of reactions? Which means rate of reaction means according to enzymes, amount of substance changed in a given period of times, or amount of substance changed into products in a given period of time. That period is called rate of reactions. The amount of substance that converting into product in a given period of times is called rate of reactions. We have a variable or environmental condition affecting the rate of the reaction. One is temperatures, pH, that's study about acidity and basicity of substance, substrate concentrations, enzyme concentrations, inhibitors and active enzyme activators. Those who release non-competitive inhibitors. When we see one by ones, temperature is, or if enzymes at very low temperatures, enzymes become inactive or it's not catalyze reactions. Enzyme activities increase gradually when the environment or temperature in the environment is rise in temperatures until it becomes at optimum temperatures. Each enzyme has its own optimum temperatures. Optimum temperature means the temperature at which enzyme shows its maximum activity. The activity that enzymes shows its maximum temperature at which enzyme shows its maximum activity is called optimum temperatures. For human beings, our optimum temperature is 37 up to 40s. For thermophilic bacteria or the bacteria that live in volcanic eruption is 19 degrees Celsius. 90 degrees Celsius. So the temperatures above optimum temperature and below optimum temperature is decrease enzyme activity or denatures the structure of enzymes, especially active site of the enzymes. Active site of the enzyme. If this is optimum temperatures, if the temperatures rise, the enzyme activities become decrease. Once enzyme is rise, it reaches at optimum temperatures. When enzymes denature, their reactions become decrease. So generally, the pH value is it's valid about acidity and the basicity of solution of that enzyme is act on. If that solutions have lower pH value, which means it have higher hydrogen ion concentration and lower hydroxide concentrations. Enzyme is affected by changing the pHs, which means pHs affect charges of amino acids because if enzyme is proteins, enzymes have amino acid inside. So the charges of pHs affects charges of amino acid that's found in active site of an enzyme. So the property of the active site change, substrate can no longer bind. Properties of active sites determine binding and unbinding of substrates. The most favorable pH value, the point where enzyme is most active, is known as optimum pH value. The most favorable pH or the most pH where enzyme is most active is called optimum pHs. Each enzyme has its own optimum pHs at which it attains maximum activity. Optimum pH means the pH at which enzyme is attain or work its maximum, shows its maximum activity. For most enzyme, this is about pHs about 7 up to 8 is the normal body pH of the enzymes, but few enzymes can work at extreme pHs. The enzymes that work in low pHs or acidic pHs like gastric protease or pepsin in our stomach, which has optimum pH is 1. The most acidic enzyme working enzyme pH 1 is pepsin or gastric protease enzymes. The third factor is concentration of enzymes. An enzyme concentration increase, the rate of reaction also increase. If we are increasing the concentration of enzyme in the reactions, he will increase the rate of the reactions. However, this will only have effect upon certain concentrations, where enzyme concentration is no longer limiting the factors. Because more enzyme molecules available to catalyze the reaction, more enzyme substrate complexes are formed. If we are adding more enzymes to the reactions, we have more enzyme substrate complex are formed, which means it have positive effects on the reactions. The factors. The another one is substrate concentrations. The concentration of substrates also have its own impacts on the rate of the reaction of enzymes. It has been shown experimentally that if the substrate concentration is increased, the reaction velocity will increase until it reaches maximum substrate enzyme binding. The reaction velocity or Vmax is increased. This means because more substrate molecule can collide with active site, more substrate is bind with the active sites. Therefore, more enzymes or more enzyme substrate complexes formed. More enzyme substrate complexes formed. More enzyme substrate complexes formed. Which at by Vmax or maximum velocity, the enzyme is completely saturated with substrate. Enzyme is completely saturated means enzyme completely bind with substrate. All enzymes bind with substrate. Enzyme is saturated means enzymes bind with the substrate. After reach a maximum increase in substrate after reach a maximum, once the substrate and enzyme concentration is reached a maximum, and if we increase in substrate concentration, it will not increase the velocity because there's all substrates bind with enzymes. There is no left enzymes. The reaction is reaching a maximum. If you are adding a lot of substrate or increasing substrate, it will not increase the reaction velocity. At higher concentration, the enzyme molecules become saturated with substrate. If you are adding enzyme molecules, enzyme is saturated with substrates. And there is few free active enzymes, which means adding more substrates does not make difference on the reactions, which means does not speed up of reactions of that concentrations. This is its own impacts. Let me see or understand by these graphs. We have substrate concentrations here. We have rate of the reactions. We are adding substrate, the graph is increase. Then it reached at a V max. V max means the maximum that enzyme is catalyzed. Then if we are adding another substrate, all enzyme is bounded by active site or saturated. There is no free enzyme, so the reactions remain the same if you are increasing substrates. So it's its own effects on the enzymes activities. And also the another factors affecting enzyme is enzyme inhibitors or presence of inhibitors in the substrates. Inhibitors means a substance that hinder substrate binding to active sites. It prevents substrate from binding to active site. Maybe it competes with substrate, maybe attached with allosteric site of the enzymes. Enzyme inhibitors are a substance which alter the catalytic action of the enzymes. And they consequently it will make what? Slow down the reaction of the enzymes, or it stop the catalysis of the enzymes. They have great impacts. Inhibitors is slow down the reaction of the enzymes or stop catalysis of the enzymes. Then we have different example of inhibitors. It's very important, or poison for human beings. Nerve gas, snake bite, cyanide, heavy metal. Many drugs are inhibitors of enzyme in nervous systems. We have two types of inhibitors. The first one is reversible inhibitors, competitive and non-competitive inhibitors. But these competitive and non-competitive inhibitors is class of reversible inhibitors. In grade 11, this reversible is divided into two: competitive reversible inhibitors and non-competitive reversible inhibitors. Let me see one by one. Reversible versus irreversible inhibitions. Reversible means once it is bound with the enzymes, it may be loosed from the enzymes or changed or released from enzyme active sites. Many enzyme inhibitors is bind to the enzymes by weak bonds, in which its case inhibition is reversible. It's bind with the enzymes by what? Weak bonds. Reversible inhibitors means the inhibitors that bind to the enzymes by weak bond for temporary, not permanent, for temporary or short period of times. Some reversible inhibitors resemble normal structure molecules and compete for admission into active site. It's compete for active sites. Compete with substrate to bind with active sites. It can be overcome by increasing concentration of substrate so that active sites become available. If we are increasing concentration of substrate, the active sites become available. Active sites become available. The increasing concentration is solution for reversible inhibitors. Irreversible inhibitors usually covalently modified an enzyme, and the inhibition cannot be reversed. It is bind to the enzymes by covalent bond or by a strong bond, and once it is bind, it's not reversed, it's not released from the enzymes. It's reactive the functional group or active site of the enzymes. This irreversible inhibitors is not contain reactive functional groups or inactivate enzyme activities. It make enzymes inactivations or totally block the enzyme's activity. Irreversible inhibitors are generally specific for one class of enzymes, do not inactivate all proteins. This reversible inhibitors is generally specific for active site of the enzymes, do not inactivate all protein parts of the enzymes. Reversible inhibitors do not function by destroying protein structures, but specifically altering active site of the enzymes. So generally, these irreversible inhibitors block the active site of enzymes, not general parts of protein structures. It is block active site of the proteins. It is block the active site of protein enzymes, not another protein structure of the enzymes. When we come to competitive inhibitors, occurs when inhibitors has similar structure to the substrate molecules. Competitive means those who compete with something. These competitive inhibitors compete with substrate bind to active site of enzymes. These inhibitors have similar structure with substrate, similar structure to substrate molecule. So it can feed or bind to active site of the enzymes. So it prevents binding of the actual substrate. When these competitive inhibitors bind to active sites, it prevents binding of actual substrates. Then the reaction is inhibited. The reaction is blocked. Therefore, it's compete with substrate for active site. It means it is slower down the reactions or block the reactions. The common example of this competitive inhibitors is penicillin. That's a drug for using for antibiotics as antibiotics for blocking bacteria. It is block the cell wall of bacteria, construct their bacteria. The enzymes that produce cell walls is blocked by these penicillin inhibitors or drugs. Increasing concentration of substrate is restore reaction rate, and inhibition is usually temporarily and reversible. So this competitive inhibitors is reversible inhibitors and also temporarily binding with active site of the enzymes. It may be restored. Our enzymes become restored and we are using again. The solution is increasing the concentration of substrate. Then we restore the reaction rates and we're releasing inhibitors. We reverse it. We have inhibitors, we have substrates. Then if this inhibitors is bind first, the reaction is blocked, or substrate is prevented binding to the active sites. You see, it is prevent the bindings of the inhibitors, substrate, sorry. The second one is non-competitive inhibitors. It's another example of reversible inhibitors. These inhibitors is occurs when inhibitors have different structure from the substrate. Non-competitive inhibitors means it's not compete with substrate. It have no structure, similar structure with substrate. It do not fit to active site. It's not have similar structure with active site, but bind to enzymes at location other than active site. Do you see the difference between competitive and non-competitive inhibitors? This non-competitive inhibitors is not bind to active site. It bind to location. It bind to location other than active sites. You see, it is different in structures. But when we see competitive, it is similar structure with active sites. But this non-competitive is not fit to active site, but bind to enzyme at a location other than active site. The location other than active site is called allosteric site. In some cases, this inhibition is thought to bind to enzyme in such ways, physically block normal active site of the enzymes. In other instances, binding of these inhibitors is changing shape of the whole enzymes, including active site, and preventing it from reacting with substrates. So, these latter types of non-competitive inhibition is called allosteric inhibitions, and also the site where inhibitors is bind to the enzyme is called allosteric sites, and also the processes inhibition is called allosteric inhibitions. Tend to bind tightly. It binds with the enzymes tightly and by strong covalent bonds. It is reversibly. It's also another example of reversibility. Therefore, reduce the amount of active enzymes. For example, cyanide in our digestive systems, peritoxin, heavy metals in our bodies have its non-competitive inhibitions. This inhibition is bound to allosteric site of enzymes. Instead of active site, it's bound with allosteric sites. It's bound with allosteric sites. This changes conformation of active sites. It is change conformation of active sites. You see, it changes conformation of active sites. Substrates no longer fit with properly. It's not stay with active sites. It's released from the active sites. These non-competitive inhibitors have its own advantage or function in metabolic pathways. Allosteric effectors or feedback, we call feedback inhibition or end product inhibitions. This non-competitive inhibition is called allosteric inhibitions. Allosteric inhibitions. The place where inhibitors bind to enzyme is called allosteric sites. A place where inhibitors is bind to enzyme is called allosteric site, not active site. Be in the mind. You must be in your mind. Competitive inhibitors is bind with active site, but non-competitive inhibitors is bind with allosteric site. Inhibition of an enzyme by product of its pathway is form of negative feedback. When the last product is inhibits the first reactant, it is called negative feedback or end product inhibitions. The activity of some enzyme is controlled by certain molecule binding to specific regulatory site known as allosteric site known on enzymes. Binding to enzyme allosteric sites. It's controlling enzyme activity inside living cells. The molecules that bind to allosteric site is called can either inhibit or activators. Such enzyme is called allosteric enzymes. They often occur at the start of long biochemical pathway in our cells. It is activated by the substrate of pathway and inhibited by product of the pathway. You see, the reaction is activated by substrate and also inhibited by products or end product inhibitions. It's only turning pathway on when it is needed. The pathway is turned on. Pathway is on when it is what? Needed. This process is called feedback inhibitions or end product inhibitions. End feedback inhibitions or end product inhibitions. The last one is enzyme activators. When enzyme is inhibited by non-competitive inhibitors, we have released these non-competitive inhibitors by activators. The allosteric control can involve in stimulation of enzyme action as well as inhibitions. Once enzyme is inhibited, we are stimulating the enzymes by these activators. We are stimulating the enzymes by the activators. An activator molecules can bind to allosteric sites, can bind to the allosteric site of the enzymes, then induce the reactions at active site by changing its shapes. The active site of the enzyme is change its shape and correct itself, and also feed substrate. It could not induce change by itself. The common example of activators is hormones in our human cells, different organisms, and is a product of earlier enzymatic reactions. Allosteric stimulation and inhibition is allow production of energy and materials by the cell when they are needed. It inhibits production when supply is adequate. Competitive inhibitors inhibit the reaction when the reaction is adequate or enough for our cells. So this is all about metabolism and also parts of enzymes. This is double education channel of educations. This is your channel. It's prepared by your teachers. You will learn from this video a lot of things, I hope. But what you do for me is, please, if you are new for my channels, like, subscribe, and share for your friends. Never forget. If you are my clients, please like my video. If you have any comments, if you have any topics that's present, I will prepare for you. Put on the comments. Your comments is give me energy for me. See you next class. Love you. Please. Thank you for watching double education channels. Please subscribe my channels and also share for your friends. I love you to all of you. See you in next class. If you have questions, suggestions, comments, put under the comments of my channels.